Hello friends, again this is Neha Antal, an assistant professor from IMS Katsabat University Courses campus. Now we will continue the bacterial staining topic and let this topic take it further. Now we will study acid fast staining. What is acid fast staining? It is a special staining as it stains those bacteria which does not take simple staining. This staining, firstly we introduce this stain as a differential stain as it differentiate two different types of species of bacteria. It was developed by Paul Ehrlich and later on modified by Jill Nansen. It is generally used to study or stain mycobacterium species, especially M. tuberculosis, which is the causal agent of a tuberculosis. It is having high lipid content, which makes decolorization very difficult. This is it. This is extraordinary property of mycobacterium. One principle, as we have discussed, property of mycobacterium species as it is having high lipid content. If once this bacteria is stained with primary dye, it is difficult to decolorize with acid. This property is due to the presence of mycolic acid in their cell wall. Mycobacterium tuberculosis, which is the causal agent of tuberculosis, it's also an alcohol fast. Staining reagent, as we need some staining reagent in gram staining. Likewise, here also staining reagents are there. First is strong carbon fusion, which is used in again primary stain which gives color to the bacterial culture. Then we will use 20% sulfuric acid, or we can use 3% HCl, which will act as a decolorizer, means decolorize the stain if it has a possibility to lose the color. It is having acid fast property. After using HCl or H2SO4, we will use 95% alcohol, which is again used to act as a decolorizer, and this is a alcohol fast property. And last one, we we use methylene blue or malachite clean, which is used as a counter stain. If the bacterial culture uh, does not take or lose the primary stain, then they will take this methylene blue or malachite green color. No, one special note is there. If we are going to study Mycobacterium leprae, which is the cause of leprosy, in second step we will use 5% sulfuric acid, and if we are going to study Nocardia species, then we will use 1% sulfuric acid in second step. Procedure. First we have to prepare the smear. Now you easily know how to prepare a smear. Then we will add strong carbon fusion. Then after adding carbon fusion, we will heat it till steam rises. When we see the steam, we allow 5 to 10 minutes to act, or we will wait. Then after, we will wash the slide. After washing, we decolorize this with acid alcohol mixture till we'll get a faint pink color in the smear. Here we will take 3 to 5 minutes, and after that we will wash it again. After washing, we add methylene blue or malachite green. We will retain it for 2 minutes and then we will wash again. We will heat and dry it, allow to dry, that is air dry, and focus under microscope and we'll study. Result. When we study it under microscope, there are two types of colony we are able to see. If the bacteria is appearing pink in color, it means they are acid fast bacteria or bacilli. Example, Mycobacterium tuberculosis, which will look like long cylinder bacilli, or a Mycobacterium leprae, that is short thick bacilli. If we are able to see blue colored bacteria, we are seeing a culture which is having a blue colored bacteria, it means they are non acid fast bacteria because they didn't retain they they didn't retain the primary stain, and that's why they are appearing blue in color. Example, epithelial cells, pus cells, and other bacteria. The result will look like this, like you are seeing here. This rose shaped bacteria, long slender thing like structure, they are retaining primary stain due to the presence of high lipid content in their cell wall, that is mycolic acid. Here is the result final. This is how mycobacterium looks like after the culture. Thank you.